there's multiple places that have joined this collaborative effort to make therapy, to make these therapies a reality. And I think that's really what's really exciting about the scientific, community, is that, they're able to kind of come together and make these therapies, a powerful option for patients in the future. Yeah.
This is the James Cancer-Free World podcast. I'm Steve Wartenberg, and my guest is Dr. Richard Wu, an MD and PhD. Richard is a medical oncologist. He specializes in skin cancer and melanomas and the early development of new drugs to treat these very common forms of cancer. And that's our topic today, a very new and promising treatment. It's called tumor-infiltrating lymphocytes, or TIL, and it's a treatment that's so new. I really couldn't find very much information on it as I prepared to talk to Richard. So I'm very curious and excited to learn all about TIL and the impact it's going to have for cancer patients. Welcome to the podcast, Richard.
Yeah, thank you for inviting me. Yeah, glad to be here.
Yeah, before we jump into TIL, I'm always curious, what was your motivation? What drove you to become a MD and PhD and specialize in oncology and skin cancer and melanoma?
As I was growing up, I started to kind of see that some of my family members were, suffer, from cancer, and some of them really suffered toward the end. And I remember one of my aunt, was treated for I think it was for cervical cancer, and it was advanced, and she participated in a clinical trial at MD Anderson at the time, but she didn't, but the therapy didn't work out for her. So I think seeing how much that they suffer, that really motivated me to research, to find new ways of tackling this terrible, afflictions.
Well, I was going to say, you're far from the first doctor I've talked to who a situation in their own family.
Yeah. So when I first, joined the NDPH program, which was done at one of the top tier cancer center in the nation, which is MD Anderson Cancer Center. That was a time when immunotherapy actually wasn't popular at all. At the time, there were a couple of biotech companies that had developed a slew of cancer vaccines, and at the time there was a lot of hype of that maybe cancer vaccines will be able to cure cancer, but then most of these cancer vaccine trials sort of flopped. So at the time, people really weren't interested in cancer immunotherapy, and for me, I sort of took a leap of faith. I thought that this idea of using your own immune system to attack cancer, seems a very interesting idea.
Okay, so let's now jump in. We've got your background and your motivation and how and why you got here. And so let's talk about, should I call it TIL? Is that the accepted name now?
Yes. So the full name is actually called the adoptive T cell therapy using TIL. That's really the way I would describe it. Yes.
Well, let's start with that word lymphocyte, because that seems to be the most important word, and I know a little about them, but let's start with that. What are they? And I know they're part of the immune system. So what role do they play, and what exactly do you do to them in the TIL procedure?
Yes. So I like to kind of think in terms of, like a common scenario. Let's say if someone gets like a flu vaccine, get a flu vaccine, right? I mean, how does it, how does the flu vaccine work? Because, by sort of knowing how that works, then we get an insight into how the immune system sort of help us, develop a response to future infections. So what flu vaccine is essentially a protein that's taken, that's a fragment of the flu virus, and so when a patient gets a flu vaccine, what happens is that your immune system sort of takes up that little fragments, and it's taken up by a type of cell called dendritic cells, which we call the antigen presenting cells. So what they do is that they take up these fragments of the virus and then sort of alert the part of our immune system called the adaptive immune system, which are essentially consisted of these immune cells, such as something called T cells or B cells. So essentially, these dendritic cells, they're like a trainer. They essentially educate our T cells to or B cells to recognize these fragments of a virus, so that when so that if a person is exposed to the virus in the future, then the immune system can quickly recognize it and kill off the virus. So that's essentially the function of our T cells. It's a part of the immune system that's very specific, very good at recognizing threatening like signals or dangers that can be that can rise from either virus or cancer.
So they recognize things in your body that shouldn't be there.
Yes, exactly. Yeah.
Okay. Now, T cells, are they the same as lymphocytes, or different, or connected in what way?
Yeah. So T cells is it's essentially lymphocytes. I mean, we use either term interchangeably. Yeah.
Oh, okay, got it. So until you're taking, well, you describe this, what do you do to the lymphocytes to make them more effective?
Yes. So what happened is that that in cancer patients, particularly melanoma patients, which is the type of patient that I treat, if you were to take out a piece of your tumor, you will see that oftentimes that they're infiltrated with lots of immune cells, especially T cells. But for some reason, some patients they're not able to, the tumor doesn't shrink, and I think part of it is because within the tumor itself, there's a way for the cancer cells to shut off the immune system, probably through a molecule called PD-L1. So the idea with the TIL therapy is that is to take these cancer fighting T cells away from the suppressive environment within the tumor, and so that we'll have an easier time of growing them out in a laboratory. And the good thing about these T cells is that the reason that they are there in the tumor in the first place is because they recognize something, some signal from the tumor, and that's why they are there. But for some reason, they are not able to kill off the tumor. So by taking them out and grow them in the laboratory, maybe we can remove them from this suppressive environment and make them potent again.
So, okay, let me make sure I understand this, because this PD-L1, I've heard this before. This is some sort of protein that it affixes itself to the outsides of the cancer cells and makes it so the T cells sort of glide by without thinking anything's wrong. And that's a lot of immunotherapy is targeting PD-L1 right now. That's like one of the main targets of immunotherapy. So you're doing the same thing but you're actually taking the cells out of the cancer tumor.
Yes.
And those are the ones you send to the lab to genetically modify. Is that the correct term, process?
So this is different from the CAR T therapy. So CAR T therapy is something that we use for the blood cancers, like leukemia, like
right.
Lymphomas. For the so CAR T is different from TIL because CAR T these cells there, these T cells are engineered to only target to only hit one target on the cancer cells. And I think in the case of CAR T, the CD19 is a very common target that's used for that. With the in a solid tumors, oftentimes they have multiple targets. They don't only have they don't just have one target. They have multiple sort of something called antigens. Meaning that it's a fragments of a tumor cells that's generated from their mutations. And so in a solid tumor, sometimes you can see like thousands of them. So in that setting, the CAR T really doesn't work because CAR T can only hit one target, whereas in solid tumors you have multiple targets. And so the advantage of a TIL therapy is that the T cells that's infiltrating these tumors, they are responding to these multiple sort of signals from the tumor itself. They're trying to hit these multiple targets, but they're not able to do their job because of suppressive tumor microenvironment. So by taking them out and growing them in a laboratory, that's one of the ways that we can make them do their job as a cancer killer. Yeah.
Now, if I understood you correctly, did you say grow them in the laboratory? Is that?
Yes.
So what does that mean? Grow them, like make them multiply more than they normally would, or something different?
Yes. So oftentimes when we take out these tumor cells, the T cells from the tumor, the TILs, we start out with maybe like a couple millions of cells, and then we put them through a special procedure called the rapid expansion protocol. And I often abbreviate it as REP.
Okay.
And so basically in this protocol, now these cells that are growing in the laboratory, they're exposed to very potent stimulus to make them grow and regain their function. And as a result, these cells, they can expand 1,000-fold to billions of cells.
Wow, that's amazing. Now we're going to take a quick break, and when we come back, Richard, you'll fill us in on what happens when you put these T cells back into the tumor. In today's world, misinformation abounds, but at the Ohio State Health and Discovery website, we're addressing today's most relevant health, wellness, science, and research topics, all from the Ohio State experts you can trust. We're tapping into physicians, scientists, and thought leaders across our medical center and health sciences colleges to give you the deeper story behind the headlines and the truth about the topics affecting the health of individuals, society, and the world. Visit health.osu.edu today. We're back with Dr. Richard Wu, and we're talking about tumor-infiltrating lymphocytes, or TIL, this amazing new process that Richard just described, in which T cells are removed from inside a solid tumor and then multiplied by 10, 100,000-fold in the lab, and then injected back into the patient's tumor. And so what happens, Richard? First, how do you get them back directly into the tumor? And then what happens when all these lymphocytes, or T cells, billions of them, are there in the tumor?
Yes. So just to correct this a little bit. So what happened is that when these TIL products, they're generated, they're billions of cells, and so oftentimes the product itself is going to appear in IV infusion bag. So before we infuse these cells back to the patient, there's a procedure that would put them through. It's something called lymphodepletion. So what happened is that that in patients' bodies, they have these multiple sort of immune cells that can potentially suppress its activity, and they can also potentially compete with these infused T cells. So it's important that we do this procedure called lymphodepletion, where your patients are treated with about like 5 days of chemotherapy to help deplete out these suppressive cells, and also to kind of reduce the existing immune cells, so the new cells have space to grow.
Oh, okay. Creating space for them.
Yeah.
Got it. Okay. And then what happens when they're back in? What do they do?
Yes. So then after the lymphodepletion procedure, which will be done in an inpatient setting, that's when the patient's body will be ready to receive these manufactured TIL products, and that's essentially their own T cells that have been expanded 1,000-fold, and the people have shown that they can specifically recognize and home into a tumor itself. And because they have been grown in the way that sort of maximizes their tumor killing potential, these cells are actually very different from the cells that were initially isolated from the tumor itself. And so after patients had after they get this infusion of the TIL cells, we then give them something called a cytokine, something called a growth factor, that sort of helps these cells grow. It's called IL-2, and these IL-2 will be able to help sustain these cells once they're infused back into the patient.
And when they're infused back in, they recognize and kill cancer cells. Yes. And do they continue to keep multiplying and growing? So you sort of have an ongoing supply of these multiple great killing cells.
Yes. So I think this is where this rather personalized nature of the therapies will come into play. So I think in my experience of having done research on these therapies, we know that every patient's TIL product can be a bit different. Some patients TIL can last longer, and some patients TIL may not be able to last as long. And we also have reported that the ability of these cells to persist after infusion, strongly correlated with clinical response to these therapies. And right now, there's ongoing research effort to try to discover ways, is there any signals on these T cells that we can manipulate to make them last longer?
At what stage are you in this, in the clinical trials and the FDA approval process?
So, so the company that has pioneered these TIL studies is a company called Iovance, and I think they're really the furthest ahead in terms of development of this therapy. And from what I heard, is that they have that they are waiting the FDA's decision to approve these therapies for the patients for the melanoma patients that have previously failed the standard immune checkpoint therapy, such as like Keytruda or Opdivo, those therapies that were commonly used for melanoma. And they have presented data that in these difficult to treat patients, if they are treated with the TIL therapies, sometimes we can salvage response in about a third of patients, which is probably the best among all the therapies that's available right now in that setting.
Okay, and about a third, because already an X percentage of patients have responded well to the traditional immunotherapy, and for those who don't, another third are going to respond well to this.
Yes,
okay.
and I also want to highlight that for patients that currently, for patients that don't respond well to the standard immune checkpoint therapies like Keytruda or Opdivo, their prognosis generally is not good. So that's why in that population, there's a need to find new approach to treat these patients. Yeah.
There would be very, very limited options for them, with TIL being the best one, it sounds like.
Yeah, currently, yeah, I think so. Yeah.
So if this company is seeking or close to FDA approval, then clinical trials have been held around the country and at the James.
Yes, actually, for the over the past probably more than a decade or so, it's really interesting to see how this therapy was was developed, and it was only done at the National Cancer Institute, and now over a decade or so, it has spread pretty much throughout the world. I have had colleagues at Toronto that they were part of this study as well, and then and also colleagues at I'm office cancer in Florida, and a couple Sloan Kettering and the Anderson. I mean, there's multiple places that have joined this collaborative effort to make therapy, to make these therapies a reality. And I think that's really what's really exciting about the scientific community. Is that they're able to kind of come together and make these therapies a powerful options for patients in the future. Yeah.
Now, to me, it's interesting because we've had a couple people come on and talk about CAR T cell, and that's for blood cancers, leukemia, lymphoma, and they've always talked about how the next step is to find a similar type of treatment for solid cancers, for melanomas, lung cancer, head and neck cancer. This sounds like a tremendous possibility to be that solution equivalent to CAR T cell, but for solid tumors.
Yes,
and
exactly. Yeah.
And that you here at the James and other places are really leaders in this.
Yes. And also, I want to just emphasize that I think this is only the tip of the iceberg. So I like to kind of call this TIL therapy like a platform technology. It's I mean, we're still at the very beginning of the TIL therapy, and there's still multiple ways that are being explored by the research scientists to see how we can make this therapies even better than it already is.
Yeah, that's what I wanted to kind of end with. If you look ahead 5, 10, 15 years from now, and is how long it takes to really perfect and fully understand these new kind of treatments, what do you see? What types of cancers and what types of responses do you think can happen if things work, hopefully, knock on wood, the way you think they will?
There's ongoing trial in I think with cervical cancer, in lung cancer, in head and neck cancer. and hopefully, there'll be more studies exploring other solid tumors too. I think efforts are certainly very ongoing in that regard. And actually, one of our investigators here is involved in the TIL study for the lung cancer. So so I think it's going to be it's going to be very exciting the next few years. When we start to see more readout from these clinical trials of TIL therapies that's used in other cancer types. And also, we're we're also trying to explore new ways to make this therapy work better. I know that Alvance has an ongoing study, to try to take out the PD-1 molecules on these TIL cells and see if they can work better, right? Because right now, we know that that the cancer cells, they express this molecule called PD-L1, which can shut up these immune cells through a molecule called PD-1. So what happens if we just remove the PD-1 molecules on these TIL cells? Will they work better? And so I think the OSU is actually one of the participating site for that study as well. And we're definitely looking for patients, to be part of a rather exciting study here. And for my clinical research effort, I'm also looking at the application of a TIL therapies at an earlier stage of disease. So the rationale is that for stage 3 patients, before the disease gets to stage 4, can we give them the TIL therapies, which will potentially give them a much longer, durable response. So essentially, they will just get treated one time, and hopefully they can be cancer free for a very long time too. So these are some of the exciting directions that we're exploring. Yeah.
What's it like to sort of be at the forefront of a whole new form of treatment and be able to have options for your patients that you wouldn't have had 5-10 years ago?
I think that's really what I do. It's really what drives me, in my daily work. I've seen patients that don't respond to the standard treatment options that we have right now, and I think seeing their suffering, really kind of validates my desire to come up with a new approach, right? I think we should always be pushing the frontiers. We're never satisfied with the status quo, and I think that's what really I think patients, they're really a part of what drives me to dedicate myself to this clinical research effort. Yeah.
Yeah, and I think that, as you started off telling us, it goes back to your own family experiences.
Yes. Yeah. I think cancer is a terrible disease. The hope is that maybe one day we can make this a more manageable disease.
Well, thank you for sharing this exciting TIL therapy, and perhaps down the road, you'll come in and fill us in on the new advances.
Yes, definitely. Yeah. Thank you for inviting me today.
My pleasure. This podcast is brought to you by the Ohio State University Comprehensive Cancer Center, Arthur G. James Cancer Hospital, and Richard J. Solove Research Institute. For more information, check out our website, cancer.osu.edu.